In this video, we will discuss about this Oncolytic Herpes Simplex Virus remodels the tumor microenvironment. So before this paper, people know that there is Oncolytic Herpes Simplex Virus or oHSV remodels to TME. But what are the changes that it does is not been studied in detail. So this paper does a detailed study of how this oHSV affect this TME. So in the cold form is the conditions where it is not treated with the oHSV, and the hot form is the conditions where it is treated with the oHSV. So why they are called cold and hot, we will understand this thing. So before that, what kind of model they use, that is high cancer associated fibroblast enriched tumor microenvironment. So these models that they have generated using the 1:10 of this KPC, pancreatic adenocarcinoma, and this pancreatic stellate cells. So its ratio is 1:10 that they have used to make this cancer associated fibroblast enriched tumor microenvironment. So what they are able to find out, when they infect the when they induce this oHSV, then the tumor microenvironment subtype transformation occurs. In these subtype transformations, if we go more into detail, what happens is this myofibroblast that helps in the immunosuppressions goes down, and the antigen presenting cancer associated fibroblast goes up. And some of the antigen presenting antigens that are mentioned in the abstract are this major histocompatibility complex 1, MHC-II, and CD86 that helps in the detections of these tumors, or in this case the tumor microenvironment. And because of that, these antigen presentations, it will lead to the T cells activations, because MHC1 and CD86 are because the interactions of CD86 and MHC1 can lead to these T cells activations, and once the T cells gets activated, it will attack on this TME and the tumors that will release through this release of the chemokines, and these chemokines will then recruit the other immune cells such as this macrophages, and this immune cells has a role to release this nitrogen oxide, and this nitrogen oxide release thereby helps in this tumor inhibitions. So these are like three major aspects where this oHSV infections works. So now let's come to the cold. In the cold case, the there is immunosuppressive environment is very strong. It can be seen here, where it is surrounded by this cancer associated fibroblast. Myofibroblast is generated through cancer associated fibroblast, whereas in the case of this induced CAF, where it is induced by this oHSV, we can see that lots of this oHSV, and at the same time there is a decrease in the myofibroblast, that is cancer associated fibroblast, that has this higher composition of myofibroblast goes down. At the same time, what happens is this antigen presenting cancer associated fibroblast goes up, and because of that, we can see there is a release of IFN gamma, which is also helps in the immune cells activations, and also there is a release of nitrogen oxide that will prevent these tumor inhibitions. So overall, the abstract shows that there are three major ways by which this oHSV works. One by subtype transformation, that sometimes even leads to these differentiations, where tumor suppressive cancer associated fibroblast goes down, and the immunosuppressive cancer associated fibroblast goes down, and the tumor suppressive cancer associated fibroblast goes up here. And on the other hand, because of that subtype transformation, antigen presentation improve, the immune cells will be able to detect the antigens, tumor antigens or tumor associated antigens, and thereby it will lead to the release of chemokines, cytokines, and that will recruit more macrophages and also the neutrophils. Neutrophils, I forgot it before. And this macrophages and neutrophil has the ability to release nitrogen oxide that will help in the tumor inhibitions. So this is what the abstract says. We will discuss in detail in our subsequent videos the different figures that are presented in the main paper and how they come out to the conclusions that are mentioned in the abstract. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.